It's my pleasure to introduce also the next speaker, Dr. Colwyn Headley from Stanford University, who is going to tell us about rescuing aging-associated immune dysfunction via mitochondrial transplantation.
So thank you for the invite and the opportunity to share some of my, crazy science. And I'll get the acknowledgments out of it before I start because it takes a village. But anyway, so I'm going to talk about the immune system, and I'll use analogies because I don't expect everyone here to be an immunologist. But this is like a healthier young immune system. You've got great gas mileage, brakes work, you can weather the storm, you can survive a crash. But as you get older, or if your immune system kind of resembles a old car, a clunker, high mileage, horrible brakes, engine failure, you're gonna need insurance for sure. So this is really what it's a good analogy to explain what happens as you get older. There's your prevalence rate for developing chronic pathologies increases, and it's been known for a long time. We've been trying to figure out why exactly the immune system doesn't work to the same propensity as the young immune system. And so, in right now, the two major camps or strategies for restoring immune function is either through depletion of specific cells or through reprogramming, targeting specific or rewiring specific pathways. And so, my science is kind of somewhere in the cut, like replacing is the strategy that I use. And so, I'll tell you today is a T cell story. So, T cell, kind of like a car as well. You need a key in the form of a cognate antigen. You need a driver as the engine presenting cell, or in this case, the dendritic cell, to activate it or turn it on. When the T cell is activated or turned on, it'll express certain markers of activation. It'll produce cytokines. It'll proliferate. So when you compare a young CD4 T cell that's versus the old CD4 T cell that's responding to the same cognate antigen, typically the older T cell doesn't display the markers of activation as well. It doesn't produce as many cytokines or the right cytokines, and there's like this cutaneous inflammation, and it doesn't proliferate to the same propensity as a young one. For the last decade or 2 decades, people have come to realize that mitochondria, powerhouse of the cell, also is like a Swiss Army knife for signaling. Like it influences every aspect of T cells as well as other immune cells. And as you get older, it's been well characterized that your mitochondria don't work as well. And so one way that people sort of measure the health of mitochondria is looking at the propensity of electrons to react with superoxide to form reactive oxygen species, which you heard about earlier. And so typically, if there's flow of electrons out of the electron transport chain to superoxide, there's a increase in ROS and a decrease in the amount of in the ability of mitochondria to produce ATP, and vice versa. So if there's a better efficiency in electron transport chain, there's better ATP production. And so you can measure superoxide or mitochondrial superoxide, and it's a proxy for how healthy a cell is. So it's kind of like the check engine light in your car. So that's what we did. We looked at the levels of mitochondrial ROS or mitochondrial superoxide in both resting or activated T cells. In both cases, we saw that the CD4 T cells from old mice, which are about 18 months, is comparable to like 65-year-old person, had a lot higher levels of mitochondrial ROS. One more quality of mitochondria I need to explain is that as you get older, there's this phenomenon called pathogenic heteroplasmy. So for a full assembly of mitochondria, 99% of that information comes from the nucleus, and then there's that 1% that's super important that's encoded in your mitochondrial DNA, the stuff that you get from your mama. As you get older, there's accumulation of mutations. For whatever reason, stuff does not stay, it's no longer healthy. And so the older you get, the more there's a shift between the quality of good to bad mitochondrial DNA that you have in a cell. And so that means is you never actually get the full assembly of a proper mitochondria the longer you live. So in real life, when your check engine light comes on, I hope you go to the mechanic because you shouldn't drive a private. You can take your car to mechanic, right? And they can put a new engine in, right? You can use that same car for another 10,000, 20,000, 30,000 if it's a Toyota for a long time, right? And so that's the principle. Why can't you do the same thing for cells in the body? And so that my hypothesis was at the time that if I can figure out how to get younger mitochondria into these old T cells, then all these things should kind of just fix themselves because of the Swiss Army knife role the mitochondria play. Just so not just metabolism, but beyond that. So what I did was I isolated some young mitochondria from healthy fibroblasts. and then I got some super old T cells, and then I put them in a centrifuge and I spun them for 5 minutes. And when I went to my mentor and I told her what I did to induce transplantation, she was like, I don't believe it. And so I repeated it with some fluorescent mitochondria just to show that they're actually going inside the cell. And so the green stuff there, those are the mitochondria after transplantation, and these are CD4 T cells. And so, pretty simple way to get mitochondria in. So to me, that was the hardest part. I was like, all right, they're in there. Let's see. Let's do some auto tuning to see how the engine works. And so when we compared the old CD4, this is a young CD4 T cell after transplantation, the mitoROS went down, suggestive of improved mitochondrial function. But we went farther and did some proteomics and some bioenergetics. At least this is what I can share for the amount of time I have today. So from proteomics, we just did a broad screen looking at all the proteins associated with mitochondrial processes. So green denotes downregulation, and red denotes upregulation. And so it's a paired experiment, O versus O plus mito. And what we see is that there's this propensity for increased proteins associated with mitochondrial processes. We put these into Go enrichment terminology, and one of the two of the major hits were related to mycose and cristae formation, so the architecture. So went ahead, did some electron microscopy, and compared the old CD4 T cells mitochondrial architecture versus that of O plus mito. And I kind of compared these to abs. They're much more protruding. Like you can see them, they're there, right? Compared to the old CD4 T cell without transplantation. So even though we saw an increase in mitochondrial proteins, a change in crystal architecture, a lot of people think that this would mean there'd be an increase in mitochondrial mass, but actually it went down. So there was some sort of reorganization and a decrease in overall mitochondrial abundance. So based off of that, we went to bioenergetics, and the seahorse is this. This is this is the bread and butter for looking at cell bioenergetics, and it's a glorified pH meter and oxygen sensor. It just measures the level of oxygen inside the media over time and pH change as cells respire. And so, based off of just a combination of different compounds, you can get like a kinetic curve of mitochondrial activity and glycolysis in the cell. And so, we looked at glycolysis, that went up, and this is again comparing O versus O plus mito. We saw glycolysis went up. We looked at oxphos activity, that also went up. So, aerobic respiration in general improved. And then, just to remind you, there was a change in cristae architecture. There was a decrease in superoxide production. So this is all happening at the same time. This is concurrent. And also, there's overall a decrease in mitochondrial abundance. So it's suggesting that the fitness, the mitochondria actually improve. It's a better engine in an old car. So the question that I always get is, okay, well, what does that mean for the functionality of T cell? So so looking at activation markers, O versus O plus mito, we saw that after stimulation, there was a better the frequency of CD69 and CD25, which are markers of early markers of T cell activation, those went up. We looked at cytokine production, so you plate the same amount of cells in a well, and then you stimulate them the same way, and the only difference is one got mitochondria and one didn't, and you can see the amount of spots. The density went up. IL-2, super important for aging, for T cell proliferation. IL-2 production tends to go down with aging. We don't know, well, we're trying to figure out why, but since we improved IL-2 production, then look, the proliferation, it kind of became predictive. Proliferation of T cells went up. So it's great. Put your mitochondria in, improve T cell activation, improve cytokine production, improve proliferation. This was all ex vivo. So whenever we go to conferences, I get, okay, Coco, well, what about inside? Put it back, put the cars back on the road. Let's see what's happening. And so we did this experiment where we took rag knockout mice don't have an adaptive immune response. So it's just if you put the T cells in there and you give them infection, you know whether the response is coming. It's based off of the T cells that you put in. So we took all naive, some got mitochondria, some didn't, just vibes, and infected them with two different pathogens. One viral, influenza, and the other one tuberculosis. And so for influenza, we looked at mortality. For tuberculosis, we looked at the CFU or colony-forming microbacterial burden in lung and spleen. In the influenza model, what we see is that the median survival went from 9 to around 12 days, significantly increased. And here are some mice with no mitochondria, no T cells at all. So you see that there's a significant improvement in this in mortality rate. When we looked at tuberculosis, what we saw is the microbacterial burden in lung and spleen, which are common indicators of, severity. The O plus mito group, or the mice that got the O plus mito T cells, had the lowest microbacterial burdens. And so, uh, quick, but so this is kind of like the research area that I'm looking at, and it's future because I don't have a lab yet. But what I'm really interested in is understanding how how the mitotransfer transfer improve the functionality of eight cells, with the ultimate, I guess, crazy goal to harness this as a way to improve human health span. And so, some examples of the things that I've done since starting that story is also looking at in human T cells. And so, elderly T cells from age range of 60, 65, give them mitochondria from fibroblasts, and then looked at two sort of terminal end states, which is exhaustion and senescence and activation. And in both cases, or I guess in all stimulation conditions, we saw that there is improved activation of the elderly T cells with decreased markers of exhaustion and senescence. So this potentially has huge applicability. But when I go to conferences, people tell me that, well, you can't put someone in a centrifuge. So, and right now the technology for matter transfer, you do a systemic injection or targeted injection, then you kind of hope the mitochondria get where to go. So it's kind of like a bus system. So I've been working on developing like a Uber, which is much more targeted. In cancer biology, there are these things called antibody drug conjugates, and so an antibody allows like the drug to have specificity based off the surface receptor. And so I've been creating these antibody mitochondria conjugates or mitotags. I think it's a catchier name, but this is just an example of what we can do. So I created this. A PCAM is a CD31. CD31 is a receptor on endothelial cells, conjugated to mitochondria, and also has a fluorescent tag. And so the idea is, if there's specificity, then there's going to be better uptake and an easier way to target delivery of mitochondria in vivo. So you don't have to take your cells out; you can just give yourself an injection. Don't give it to yourself, but so far in a co-culture model where we've got endothelial cells that express PCAM and then vascular muscle cells that don't, when we look 24 hours after just literally just spread them on the cultures, so the sort of translucent cells are the endothelial cells, the vascular muscle cells are in the green, and then the PCAM mito conjugates are predominantly within the endothelial cells. So if it's got like a almost 100% delivery or efficiency of uptake. And don't ask me why, I don't know why, but there seems to be a lot of specificity, but there seems to also be some mitochondria getting to the vascular muscle cells. And so I think that's it. Yeah, so that's the end of the presentation. But I'm happy to take questions.
Thank you so much. That was a wonderful talk. So we have time for two questions. And afterwards, we have a coffee break and we have a reception. So please make sure you ask your questions then. So maybe we start with Tim.
Hi, awesome talk. What happens if you put old unhealthy mitochondria into young healthy cells?
Would you go to McDonald's if you try to lose weight? So it's similar sort of concept. Although I didn't actually, I didn't take all mitochondria, but I took dysfunctional ones, some that were exposed to chronic like entomycin and rotenone, and they had the opposite effect of increasing mitochondrial stress in the cells. But I didn't take it farther and looked in vivo and stuff like that.
Great talk. I know you were primarily looking at CD4 T cells, but obviously when you're transferring like younger, way more active and like cytokine producing CD4 T cells, did you see any like adverse like autoimmune events or anything like that? Like when you transferred the younger CD4 T cells to older mice, or were they like still at a relative level of not being like overprimed or overactivated?
So let me go back to that. So this experiment here, these are resting naive CD4 T cells. So they haven't experienced any engine presentation stuff like that. After mitochondrial delivery, there was no avert reaction that we saw ex vivo. We injected 2 million naive CD4 into RAG knockout, and that was it. We just tracked mortality and mycobacterial burden. And so from this data, it does not seem as though there's an adverse, a detrimental impact of the transplantation and the ability of the cells to function. And the same thing with tuberculosis. So because mitochondria is, again, it's not just the ability for it to produce energy, but it's the signaling orchestra that it sort of like, mediates. I believe that's why the cells are behaving better. And we have more data that actually shows Long term, we looked at 30 and 60 days post infection and post mitochondrial delivery, and we also saw sustained lower levels of T cell exhaustion in the mice that got the mito transfer. So there seems to be some persistent phenotype. You can check out this paper, and then this paper too just got 2024, but yeah, that one just got published too, so you can see the applicability. Does that answer your question?
Okay, thank you so much, Colin. And please, let's thank all the speakers of this session for all these wonderful talks.